Hello guys, welcome to the biochemistry class. My name is Akshay. I did my MSc Biochemistry from University of Hyderabad from Department of Biochemistry, and I am pursuing my PhD from Center for DNA Fingerprinting and Diagnostics, Hyderabad. Okay, so I started this video channel to teach you guys the biochemistry. So today is my first video, and today we'll study the introduction of biochemistry. What is biochemistry basically? So biochemistry basically it is a study of biomolecules. Okay, basically in simple words it is a study of biomolecules. So biomolecules whichever we have, like protein, amino acids, carbohydrates like glucose, lipids, fatty acids, like that. So it studies such biomolecules, what are their functions in our system, how they are metabolized, structure, and all that biochemistry covers. Okay, so basically it is a study of biomolecules, biochemistry. Okay, it is a study of biomolecules. Here we will study what are the functions of biomolecules, their structure, okay, their metabolism, their synthesis and transport also it covers. So these are the different aspects of biomolecules basically biochemistry covers. Okay, so biochemistry is also called as molecular logic of life. It is also called as molecular logic of life because it studies the life at the molecular level, biochemistry. Okay, so that's why it's called as molecular logic of life. So quickly I will tell some foundations of biochemistry. First is the cellular foundation. So we'll see at the cellular level what biochemistry has. Okay, so first is cellular foundation. So as I said, cell, so we will consider cell here. So consider this is should be a better cell here. Okay, so this is the cell. Okay, this is looking good. So in cell, maybe it is a plant cell or animal cell, so it has a nucleus. Okay, in nucleus what we have our genetic material in the form of DNA. Okay, so into the nucleus attached is the endoplasmic reticulum membrane structure is attached to a nucleus, and it is a major site for protein synthesis inside a cell. Okay, and some other membrane structures also there inside a cell, like this is a Golgi complex. Okay, some other organelle like this house of a cell, mitochondria is also there. In plant cells we have one extra organelle is also called as a chloroplast. It's a chloroplast in plant cells. Okay, and there are some other peroxisomes and all that other organs. I will not go in that much detail, you guys all know that. Okay, so and rest of the here we have the cytosol, which is mostly fluidic. It has water, enzymes, inorganic molecules, proteins are there. Okay, so if you see this structure, This is the plasma membrane of cell. So plasma membrane gives the stability to the cell. Okay, so it has two major components. Plasma membrane, it consists of, one is lipid, another is protein. Okay, so lipids, they are made up of triglycerides. Okay, triglycerides are made up of simple biomolecules called fatty acids. Okay, proteins are made up of amino acids. You see the structure of amino acid. It has carbon chain, it has amino group and carboxyl group. Okay, now in plant cells we have another membrane structure called cell wall. Cell wall is made up of cellulose. Cellulose is homopolymer of glucose. Okay, homopolymer of glucose. So if you see the structure of glucose, it has aldehyde group and other carbon atoms. It has 6 carbon atoms. Okay, 1 2 3 4 5 6. Okay, it has aldehyde group. Okay, this is structure of glucose. Now next what we have our DNA here. Okay, so DNA, our genetic material, and some viruses have RNA as their genetic material. So if you see the structure of what is DNA is made up of? DNA is made up of nucleotides, simple nucleotides. Nucleotides have structures which have ribose sugar attached to a here purine ring of adenine. Okay, adenine, and this ribose sugar is attached with a phosphate. It has phosphate group as well. Okay, so this is structure of a nucleotide. So if you see, majority of important structures in cells like genetic material DNA is made up of simple nucleotide. Cell wall is made up of simple glucose. Proteins are made up of amino acids, and lipids are made up of fatty acids. So majority of cellular structures are made up of simple biomolecules. So basically biochemistry studies all these biomolecules. Okay, how they are synthesized, from where we get, how these nucleotides are converted into a DNA, how glucose is converted into cellulose, how amino acids are converted to protein, how fatty acids are converted into lipids, and triglycerides and lipids. Okay, this is how basically biochemistry covers. Now if you look at these structures of glucose, amino acids and nucleotides, you will find there are 4 common elements. Okay, carbon, nitrogen, oxygen and hydrogen. So 99% of cell mass is carbon, nitrogen, oxygen and hydrogen. Okay, and carbon has it can form 4 covalent bonds, nitrogen can form 3, oxygen 2, hydrogen 1. Okay, so and there are some other elements are also present in biomolecules. Okay, so there are 90 naturally found elements, out of which 22 are essential for life. only 22. Okay, only 22 are essential for life. Okay, so but majority of elements are carbon, nitrogen, oxygen and hydrogen. Now if you see these biomolecules, there are some important functional groups are also present in these biomolecules, which we need to know. Basic functional groups, we'll encounter these functional groups when we will study the metabolism of these biomolecules and their synthesis. Okay, so common functional groups are like hydroxyl group, here it is present, some aldehyde groups, yeah, it is present in glucose here. Some have ketone groups, okay, like fructose has ketone group. Some have amino groups, some have, like amino group is present in amino acids. Carboxyl group is present here. Some have phosphate groups, okay, some have ethers, some have esters, okay, some have phenyl groups, okay, some phenyl groups are also present. So these are some common functional groups you will see in the biomolecules. So we need to know all these functional groups. Okay, so this is how the majority of cellular structures like DNA, cell wall and plasma membrane, very important structures of cell, are made up of simple biomolecules like glucose, nucleotides and all that. And these biomolecules have some simple functional groups which are very important. Okay, that's it for the cellular foundation. So now we will see the physical foundation of a biochemistry. In physical foundation, what we will study is how the energy requirement of a cell or our system, how it is fulfilled, where we get the energy from, how we extract the energies, okay, where it is channeled. Okay, so we will study the energy requirement. So energy requirement, okay, how the cells, where the cells extract the energy, okay, where it is channeled, okay, and how it is consumed. Okay, so in basically in living system we get the energy through flow of electrons, like yes, flow of electrons provides energy. Okay, flow of electrons provides energy. Sorry. Okay, so how it happens? So if you consider a simple photosynthesis reaction, what happens in photosynthesis? Okay, the plants, what they do, they will fix the atmospheric carbon and they will reduce that carbon with the help of light energy, okay, into a glucose. Like in food, what happens in autotrophs, in plants, they will fix the atmospheric carbon, they will combine it with water and they will convert into glucose and they will release the oxygen. Okay, this is glucose. Okay, and in this process they will use the light energy. Okay, so this light energy in the form of photons is used. Okay, so the in chloroplast the light energy is consumed and it is used to excite some of the electrons in the chloroplast, and that process will go on. Those electrons will transfer and they will be given to the CO2, and this CO2 will get reduced. Okay, so this is how the reduction happens of CO2 into a glucose in plants. So what happens in heterotrophs like animals? What they do, they will do the reverse reaction. They will consume the glucose, they will oxidize, and they will release the CO2, and whatever the energy, okay, whatever the energy that basically you can say transformed from light, okay, they will store in the form of ATP, adenosine triphosphate. Okay, it is an energy currency in our body, okay, in cells. So you can see the light energy is now ultimately, you can say, it is converted into ATP. Okay, so ATP is released during the glucose oxidation. So this ATP is used to do the work in our cells. So every work, every biochemical reaction or any type of work which is done, including muscle contraction, anything, ATP is used in that process in our body. So it is an energy currency of a cell. So how this ATP is used, okay, in our system? So ATP is the adenosine triphosphate. It hydrolyzes basically into ADP and inorganic phosphate. Okay, so basically this reaction, hydrolysis of ATP, is exergonic reaction, exergonic spontaneous reaction. It releases the energy, and free energy change of this reaction is negative. It has a negative free energy change, and it's an exergonic reaction. It releases the energy. Okay, if you consider another reaction, what happens in our body? Amino acids, okay, we convert that amino acids into protein. This process is endergonic, non-spontaneous, and delta G is positive. Free energy change for this reaction is positive. So it is endergonic reaction. Energy is required to do this reaction. Okay, amino acid getting converted into protein. So in our system, how ATP is used? In our system, what our system does, it couples this ATP hydrolysis with this amino acids, with this reaction where amino acid getting converted to protein, so that this reaction will move forward. Okay, so mostly exergonic reactions are coupled with endergonic reactions in our system. Okay, so I will explain this with another example. Like you see here, this is a pulley. It is on the top of, you can say, a pyramid, a triangle. On one end of this pulley, there is a small brick attached with rope. On the another end, there is a larger brick attached. Okay, on the top, it is out the loop. So to move this brick from here to here, we have to put the energy. We have to give some energy so that it will go here. Okay, so this process is endergonic. Delta G is positive, like in this case. Now you see this brick, okay, this is at the top. It will automatically come down due to gravity. Okay, it will come at its equilibrium. Okay, so it will come very easily. So that's why it is exergonic reaction. Free energy change is negative. Delta G is negative for this reaction. Okay, so if I have to move this, I will just couple this, tie this brick to this pulley. Means I will just couple this process with this. Okay, so if I tie this here and here, if I release that, okay, break, it will also automatically come up. Okay, this is how these endergonic reactions are coupled with exergonic reactions, so that endergonic reactions will happen in our system. Okay, so this is how in our system the exergonic and endergonic reactions are combined, and the endergonic reactions and are carried out. Okay, this is how the work is done in our system. Now, as I mentioned here, some of the terms like delta G, free energy change, why delta G is negative for the ATP hydrolysis, why the delta G is positive for the this reaction, amino acid getting converted to protein. Okay, so we will see why it is there. So to understand that, we need to know some of the physical chemistry. So we will see here. So the delta G is the free energy change. Okay, and this free energy change is depend on the enthalpy change and entropy change, and that relation is given by the Gibbs-Helmholtz equation. Delta G is equal to delta H minus T delta S. This is Gibbs-Helmholtz equation of free energy. So basically the free energy depends on enthalpy. This is delta is enthalpy and delta is entropy. Okay, so what is enthalpy? We'll see what is enthalpy now. So during a reaction, a lot of changes happens in a molecule. Okay, number of covalent bonds are broken and number of covalent bonds are new bonds are formed. Okay, a lot of changes at molecular level, like vibrational, rotational energy changes will happen. So enthalpy basically depends upon these energy changes during a reaction. Like, it mainly depends on the covalent bond energy. Covalent bond. So if you consider a particular reaction, so the number of covalent bonds formed, new bonds are formed, old bonds are broken, will directly affect the delta H. Okay, change in enthalpy. Other energy changes will happen, like rotational energy changes, rotation around the molecule, rotational, and vibrational energy changes happen during reaction, like vibrational energy changes of a molecule, like molecules tend to vibrate during a reaction, and some rotational also happens. So these changes in the molecule at these levels will contribute to the enthalpy. So these are intramolecular within the molecule. Some intermolecular energy changes will also happen. Changes will happen. Okay, those are like electrostatic interactions, negative. Okay, so new electrostatic interactions formed and all electrostatic interactions are broken. This will contribute to the enthalpy. New hydrogen bonds formed, like, okay, that will also, this is the hydrogen bond between oxygen and hydrogen. So these hydrogen bonds, if they are formed or broken, that will also will lead to the change in enthalpy. Okay, so now we'll see the entropy. So entropy, what is entropy? So entropy is like if there is a randomness in the system, okay, the entropy will increase if there is increase in randomness. Like in case of ATP is getting converted to ADP, plus pi. Okay, in this case what happens, the energy of ATP now is divided into ADP and pi, so there is increase in randomness in the system. Okay, so it will increase, so delta S will be positive delta S, it will increase the randomness. Another, like amino acids, they are converted into protein. In this case what happens, amino acids, a lot of amino acids are there, they combine with each other, they will form a larger molecule like protein, larger structure protein. So in this case the energies of different amino acids now is compact to a single molecule. Okay, so what will happen here, the randomness will decrease in the system, so it will decrease the entropy, so entropy change will be negative in this case. Okay, so again delta is the entropy change will also depend upon these sort of interactions, also electrostatic, hydrogen bonds, rotational and vibrational energy changes will also contribute to the entropy change. Okay, but the only thing is different from enthalpy and entropy, that covalent bond will not contribute to the entropy change, because covalent bond is between two atoms. Okay, the energy of covalent bond cannot be distributed over the molecules, different molecules, so it is between two atoms only, it cannot be distributed, so the whatever the covalent bonds formed or broken will not contribute to the entropy. That's the only difference between enthalpy and entropy. So you can see. Okay, now we will see what happens in the case of ATP, the free energy change. So as per this equation, delta G is equal to delta H minus T delta S. So delta H is negative during ATP hydrolysis, so negative delta H minus T positive delta S, so minus delta H minus T delta S, the ultimate value will be negative, so delta G is negative for the ATP hydrolysis. What happens in case of amino acid getting converted to protein? So delta G is equal to delta H minus T delta S, so delta H is positive for this reaction, minus T minus delta S, as the entropy change is negative, so delta H plus T delta S, the ultimate value will be positive, so delta G is positive for the amino acids getting converted into protein. Okay, so this is how the free energy change for ATP for exergonic reactions is negative and for endergonic reactions is positive, like we seen here. Okay, so now we will see how exactly this ATP hydrolysis happens and how it gives a negative enthalpy change. Okay, that we will see now. So you will see the reaction here. So adenosine triphosphate, it has a ribose sugar. Okay, it is attached with a nitrogen base. NH2. Okay, and here it is attached with a phosphate. It has 3 phosphate groups. Okay, so this is the basically structure of a adenosine triphosphate. So it reacts with water during hydrolysis. I will draw water as HOH. Okay, so what happens during reaction? During reaction, what will happen is this oxygen of a water will attack on this phosphate, and this phosphoanhydride bond here, this bond will get broken down. Okay, so what it will give now? It will give adenosine diphosphate. So the structure will be same, nitrogen base, O negative, and this hydrogen will come here. Okay, so this is adenosine diphosphate, ADP, plus again what will another product. So this oxygen will do nucleophilic attack on this phosphate, and what will get? Will get inorganic phosphate like this, and that OH will be attached here. Okay, this is inorganic phosphate. So in this process, this phosphoanhydride bond is broken down, and it this OH forms a bond with a phosphate. Okay, so there are two things. One is bond breakage, that will lead to a enthalpy change, positive enthalpy change. This phosphoanhydride bond getting broken, and it will give enthalpy change positive. But again, what is happening? Another bond is getting formed. Okay, here, so delta H will be negative for that. Okay, so both the energies are equal and in opposite signs, so it will be a 0. So net enthalpy change after breaking this bond will be 0. Okay, so now you will ask me from where then the energy is coming. Okay, so it is a very misconception that this phosphoanhydride bond is it is also called as high energy bond. So it is a very misconception, because this breaking this bond is not changing any enthalpy. Okay, there is no net change in enthalpy. Okay, because there is a one bond is broken, another is formed. Okay, that will be ultimate enthalpy change will be 0. So why negative enthalpy is there for ATP hydrolysis? You can see here in this structure there are 3 oxygen negative. They are very close to each other. So there is always a repulsion, electrostatic repulsion in this molecule. So when you break this bond, there will be loss of electrostatic repulsion, and that will be a favorable condition. Okay, that will be a favorable condition. You can see the O negative is now in another molecule, and O negative. So there is a loss of electrostatic repulsion, and that is a favorable condition. That's why delta H is negative. Okay, for this ATP hydrolysis. So it is a very misconception that this is a high energy bond. Okay, due to this the free energy comes. Okay, that bond breakage will not give any change in enthalpy, but it will reduce the electrostatic repulsion. Okay, and that is more favorable. That's why we are getting delta H negative. That's the thing. So if you see the now delta G is equal to minus delta H minus T positive delta S, as entropy is increasing. So minus delta H minus T delta S, and ultimate value will be negative. So delta G will be negative for ATP hydrolysis. Okay, so delta G is negative here. So there is another misconception, as delta G free energy change for ATP hydrolysis is negative, it is a spontaneous reaction. No, in outside of the cell, this ATP hydrolysis is not a spontaneous reaction, it is a non-spontaneous reaction. Okay, why it is a non-spontaneous reaction? I will tell you. Okay, outside of the cell, ATP hydrolysis basically is an energy curve. This ATP, to reach the transition state, this is a transition state, the energy is required to reach the transition state during ATP hydrolysis. The outside of cell, this transition energy, this energy required to reach the transition state is higher. It is also called as the activation energy or and that is called activation barrier. So the ATP requires activation energy to put itself in a transition state. Okay, so that's why it is non-spontaneous. But as this ATP hydrolysis happens inside a cell and it is catalyzed by enzymes in our system, so what enzymes do? They will reduce the activation barrier, okay, activation energy. So it is enzyme catalyzed now in our cells. Okay, so that's why it is a spontaneous reaction. Otherwise, outside the cell, it has a higher activation barrier, so it is a non-spontaneous reaction. So it is a misconception that at the delta G has more negative value, so it will be spontaneous. No, it is not a spontaneous reaction outside cell. As it is catalyzed by enzymes, ATP hydrolysis, it is a spontaneous reaction. I will give an example like glucose. First step of glycolysis, where glucose is converted into glucose 6-phosphate. Here ATP hydrolysis is coupled. This reaction, glucose into glucose 6-phosphate, is endergonic reaction. So ATP is coupled with this reaction. Okay, ATP hydrolysis is coupled with this reaction, and this reaction is catalyzed by hexokinase. Okay, so that's why this hexokinase will do the ATP hydrolysis. Okay, and this endergonic reaction will move forward. Okay, so this is how the ATP is used during in our system to do the work. A lot of works are done using ATP hydrolysis, and this is how ATP hydrolysis happens. Okay, so that's it for the physical foundation. And if you like this video, please share it with your friends. If you feel it is very informative, please like the video, subscribe my channel, and I will come up with another video. So we will see you in the next video. Thank you for watching.